everyone, this is Sandamichalvi.
I am Rohini.
And now we are going to talk about the cancer diagnosis equipments briefly. Cancer diagnosis equipments is designed to detect, image or analyze the cancerous cells.
The imaging equipments are X-rays, CT scan, MRI and ultrasound. CT scans creates detailed cross-sectional images and MRI provides detailed images of soft tissues. Ultrasound detects tumors especially in soft tissues. PET scan highlights areas of high metabolic activity typical of cancer.
And then biopsy tools. Biopsy tools includes the needle biopsy devices and endoscopy systems. Needle biopsy devices is used to extract the tissue samples for analysis, and the endoscopic systems includes the visualize and collect the samples from internal organs.
Microscopes and staining systems. Microscope analyze cells and tissues. Staining systems or highlight cancerous cells in the samples.
And then molecular and genetic testing. It includes the PCR machines and NGS. NGS stands for Next Generation Sequencing. PCR machines is used to detect the cancer-specific genetic mutations, and NGS, which means the next generation sequencing, is used to analyze the DNA to identify the mutations.
Blood and tumor marker detection. Flow cytometers and immunoaccessory analyzers. Flow cytometers are identify cancer cells in the blood. Immunoaccessory analyzer detect tumor markers like PSA or CA125. These technologies are in early detection, accurate diagnosis and treatment plans.
Advanced imaging systems are including digital mammography, 3D tomosynthesis, bone scans and then fluoroscopy. Digital mammography includes the high resolution imaging for breast cancer detection, and 3D tomosynthesis is used to provide the three-dimensional imaging for better accuracy. And then bone scans. Bone scans is used to detect the bone metastasis using the radioactive tracers. And then fluoroscopy. Fluoroscopy is the real-time imaging during diagnosis procedure.
Bronchoscopy used to examine the lungs for the lung cancer, and gastroscopy detects cancers in the esophagus and stomach.
And then biopsy assistance technology. It includes the image guided biopsy, liquid biopsy and CAR biopsy systems. The image guided biopsy or the examples are CT scan, MRI and ultrasound. It combines the imaging for precise sampling. And then liquid biopsy. Liquid biopsy means the non-invasive method to detect the cancer DNA in the blood. And then core biopsy systems. It extracts the larger tissue samples.
Laboratory diagnostic tools are flow cytometry, spectrophotometers and immunohistochemistry. Flow cytometry analyzes physical and chemical characteristics of a cell, and particularly for blood cancers. Spectrophotometry measures the absorbance and fluorescence to study cellular abnormalities. Immunohistochemistry identifies specific cancer markers in tissue samples.
The next one is genomic and proteomic analysis tools. It includes the FISH. FISH stands for fluorescent in situ hybridization. It is used to detect the genetic abnormalities. And then microarray technology. The microarray technology identifies the gene expression patterns linked to cancer. And then finally the mass spectrometry. The mass spectrometry analyzes the protein and metabolites associated with cancer.
Specialized cancer detection equipments. Pap smear analysis detects cervical cancer in the woman. Dermatoscopes examines skin lesions for melanoma. Optical coherence tomography (OCT) provides high resolution imaging for skin and oral cancers.
And then emerging technologies. The emerging technologies are AI-powered imaging. The AI-powered imaging enhances the cancer detection accuracy in radiology. Thermal imaging cameras. The thermal imaging cameras are used to detect the temperature changes associated with the cancer. And then cancer breathalyzers. The cancer breathalyzers is under the research to identify cancer by analyzing the patient's breath.